Okay, good morning, everybody, and thank you all for coming. This is a meeting we've been wanting to put on for a long time. For myself, this is something I've thought about for at least 10 years, I'd say. You'd be amazed to know, actually, that one of the first meetings of this sort that is looking into this question of climate change and human evolution was held here at Lamont in September of 1984 over in the Geoscience building, which used to be our lecture hall. That was organized by George Denton, Liz Verba, and Lloyd Burkle. Probably forgetting someone else, but this is the book that, or this is the meeting that led to the sort of famous Verba volume, that blue and yellow monograph that all of you are probably familiar with. So that meeting really set the stage for what was then probably about 30 years worth of research that has happened over the intervening time. And how this meeting came about actually was the result of a meeting that was held actually in Turkana in Kenya, a meeting that was led by Richard. And he was kind enough to invite me to that meeting and even give me a special flight up because I was late. And we sat there one afternoon, and I've known him for a little while, but I'd never actually talked to him about this question. I never really got his take on what he thought about this. And so we were sitting there having tea, and Richard saying there to me to drink my water because, of course, it's hot and dry out there. And the sun is setting, and it's this beautiful sort of desert yellow glow, and we're sitting out there in the veranda and talking about it, and we came up with the idea of putting this meeting together. And so I came back to Lamont in the fall and proposed to our Climate Center, which is a seed money group, and they gave us the first little bit of support to consider putting this meeting together. And so we fast forward to today, and actually fast forward to yesterday when I gave NSF a call. As of yesterday, I had no money to support this meeting. And so you'll all be pleased to know that NSF stepped up to the table. So, I call this a symposium, and they call this a workshop. So you are now officially part of the Lamont workshop on Did African Climate Change Shape Human Evolution? There will be a product, but none of you have to do anything. I'll do it, but I will welcome your input. So one of the things I know Richard's going to call on us, really we want to make this a discussion. We want to make this, obviously, hear what people have to say, but this really should be an opportunity for people to throw out ideas and call out on things that we don't think are viable or don't work, and just really try to make a way forward. And hopefully at the end of 2 days we'll actually be able to make our way forward with some recommendations, which Rich Lane tells me we stand a pretty decent chance of actually turning into a program within NSF. So with that as a challenge, I think it's a good way for us to start. So I'd like to thank Janine Miller. Janine, you're just a miracle worker. Thank you for all you've done. And of course, there you are. Okay. So she's in the back, and she tells me, and I'm to tell you, for all of you who've traveled here, keep your receipts. Columbia doesn't acknowledge anything if you don't have a receipt to say you did something. And also to the Earth Institute, particularly Vilma Gallagher, who's around here too, I think somewhere. Thank you, Vilma. And also the director's office. Art has really stepped up to the table and helped us out with this meeting as well. So we've had funding from Lamont, from NSF, from the Earth Institute as well, in kind, from the Climate Center. And really we have a special thanks to Sarah Redlick, who has supported us in establishing the Climate and Life Institute, or the Climate and Life Center at Lamont. This is something that's just beginning now. And in fact, this is the inaugural meeting for that Center for Climate and Life. And without her support, I'd be a much more nervous person right now. In terms of logistics, we've got food and coffee throughout the day. We've got lunch for everyone. So don't go anywhere at the end of all this. We've got lots of food. We've got tons of food, actually. So, so there's plenty of food out there. Let me see, what else? Oh, we have a number of people from the media here. We've got representatives from Science and from Nature, from the New York Times, from Scientific American. Even Earth and Sky is here somewhere. So we'll be having a little radio presence today on Earth and Sky. And then lastly, for those speakers and some other people, we're having a dinner tonight that we're supposed to be there at 7 o'clock, but we'll probably leave here around 6:30 or so. Before dinner, of course, we have to have a cocktail hour, so we've got a cocktail hour at the end of the session today. So I've had my moment up here, and Richard's going to tell you all to get involved.
Well, good morning, and thank you all very much for being here. First, let me make a personal point. When I walk at the moment, my legs are loose in their sockets, so it sounds like I'm passing air. I'm not. But some of you may think, oh, effect of bipedalism. What I wanted to begin by saying is I was reminiscing a little bit with Bernard Wood, who I've known for many years, and he was saying, I was wondering what a long-lived cockroach would say if it was somewhere here and had been sort of moving around with some of us, and he'd sort of be cleaning his mouth parts and saying, same sort of people, many of them the same people. Wonder if they've changed this time. Can they now talk to each other, or are they still going to talk at each other? And that, I think, is one of the challenges that this sort of meeting has to face. When Peter and I and Lawrence Martin were discussing this at the TBI overlooking the Turkwell River last year, it seemed that there is an enormous amount of information that is beginning to come through, an awful lot of questions about how this information is being used. And I was saying to Peter that I really found many of these scientific discussions very awkward because people came with a fixed position and left with the same position because they wanted not to have to change their paper. I think you made clear there's no paper expected, so you really don't have to defend anything. And the hope is that we can actually share ideas and see where this might lead us. I get a chance to talk later on, and so I will leave some of my more sharpened remarks till lunchtime, if you like, or immediately after lunch. But as I said, what's important to me is the linkage between the work that's being done here at Columbia in their climate program, the deep sea cores, the records that are so wonderfully preserved, and what is now becoming very clear with the databases that are coming out of places like the AFAR, out of Turkana, and out of other parts of the world, where you have a terrestrial record of faunal turnover, faunal change, as well as good isotopic data on some of the levels, and to see if we can't really fit the two together in a way that makes sense in terms of understanding what is driving the changes that we're seeing, and ultimately, of course, the changes that we see in ourselves. I mean, why should evolution have produced the oddity that it has in ourselves? And I think that there is perhaps something that can be gained from this linking the whole picture and putting ourselves within the context of the changing in life with a much more accurate record than we've had to date by simply looking at the terrestrial tufa and the sedimentology that we all are hopefully quite familiar with today. So I would simply urge that in the presentations, be brief. Much of what some of you are going to say, you've probably said elsewhere and before, and I think what would be more fun is to keep it brief and really get going a good active discussion and take it in the spirit in which it's intended. If somebody says, well, can you really honestly defend that what you just said? Don't say, I'm going to go and defend it in the corner. Say, let me try. And, I think this give and take in discussion is so important, and you see less and less of it today from my experience. And so I hope that can come about. So I don't think I want to say any more at this stage. I think there's far more interesting things to do than listen to me. And I shall sneak back to my corner position and watch with interest. Thank you.
Our first speaker is someone I've known for a long time, is actually one of the people who I met at one of these very early meetings. Mikhail Fortelius comes to us from Finland, and he's been a specialist in European mammals for much of his career. And as he told me last night, he's now just sticking his big toe into Africa. And our hope is to actually get him to plunge his entire body into the problem, because he's one of the best people in the world at what he does. So we've given him the pole position for that reason. So, Mikhail.
Thank you very much. Ladies and gentlemen, organizers, thank you very much for having me. It's a fantastic pleasure to be here. I know nothing about Africa. I know very little about climate, but I have spent a long time trying to understand the relationship between what happens to mammals and what happens to their environment, and I will share that with you. I will make no pronouncements about human evolution or Africa. Very elementary question. How can we know about the past? How can we actually know what happened and why? We have started to use the term ecometrics. It's a very general term that we feel has been missing. It's really any metric you use to measure properties of past and present ecosystems. But the sense in which I use it is mostly traits of fossil organisms. Some traits that have some environmental functional significance. I will come back to this, but I wanted to start here at the interface between the mammal and its environment. The mouth, the tools in the mouth that are designed to deal with what the animal eats, that is very closely related to the circumstances of the animal. Its resource use, which depends on things such as climate, especially over long term. Climate change will change the resources available and will drive the changes that we see in this enormously rich variety of shapes available there. So there's much information to be extracted. Of course, some of this information is quite obvious and was extracted already a long time ago, such as the broad pattern of evolution of horses, where you can see the change from small animals with very low crown teeth and many toes to very large horses with only one toe and very tall teeth, reflecting the change that took place on this planet during Cenozoic time, going from more humid towards less humid and cooler conditions. So that's nothing new, but this is an example of using ecometric information, at least in an intuitive sense, as here. We can do it in a quantitative way. There are many things that could be done. I begin with an example from the group of Nikos Solounias using a technique called mesoware to look at North American horses evolving over more than 50 million years of time. The green means that they are eating what we call browse, leaves and things, and then as they get into the red, they are eating increasingly grass. This is just information from how the teeth are worn, so the direct interface between the food and the animal gives this. And I call it the long goodbye because it rather drives home the point that horses didn't leave the ancestral mode of feeding until very late. They started to expand into other modes as the planet changed, but a substantial part of the horse diversity remained where they had always been. And only at the very late stage did they actually switch. So that original idea of a rather linear change going from one state to another is actually much more complicated when you start to quantify the information that is there. It wasn't just horses, of course. It happened to all things on this planet that rely on plant foods, especially the ones that are in latitudes that got increasingly dry. And that's why today this kind of surprising situation is possible. ruminants living where almost nothing grows, not only living but growing and reproducing and feeding their owners. An astonishing ability to make living matter out of almost nothing under difficult circumstances. And in dental ecometric terms, this is why multiple groups of animals have evolved very tall teeth that can take a lot of abuse, a lot of wear, a lot of dust and sand, a lot of abrasive grass during their lifetime. So an elk or a moose, it's the same size of an animal as a horse, has only 1/10 the amount of tooth to use for its entire lifetime, and it manages perfectly well in the northern forest. But a zebra or a wild horse in the steppe or savanna wouldn't even live to reproduce with that much tooth. And this is the thing that we are looking at. This is the way that the anatomy and structure of animals is being fine-tuned to the environment, and that is why ecometrics works. Works astonishingly. Just that simple ecometric allows us to make a rainfall map of the planet today. The bottom image is the actual measured rainfall. The upper one is a rainfall map based only on the mean crown height of plant-eating mammals. See two things here. Firstly, that is a pretty good map. I mean, if we had that sort of map for the past, we'd be quite happy. And secondly, you see that there are two big anomalies. And that's where human industrial civilizations have been established for a long time. Europe and the eastern United States. So humans are having a global impact on this. But apart from that, it's an ecometric that really works. Look at Africa, how beautiful. And the time machine we use to take this kind of information back into the past is a database called the Now database that's eaten up half my professional life. At the moment it has a pretty good coverage, especially in Europe and increasingly in Asia. Africa, of course, is not nearly as well covered, partly because there is not that much information for Africa, but also because it hasn't really been in our focus. But we did this 10 years ago. We started, these are our first published rainfall maps for Neogene time, so 20 million years of changing rainfall patterns in Western Asia and Europe. I will not waste your time on these maps or on the details, but you can see that we go from humid colors, blue and green, to increasingly red and yellow colors as the planet dries, and you can see that there's quite a lot of differentiation. Not every place is changing in the same way. And indeed, if we would go into the details, I could show you examples of places going the opposite way from the global signal and speculate on why that is the case. There are regional climate effects such as monsoons that interfere with the global pattern, and we can detect them, I think. We have just done the entire northern hemisphere with this way, and there were no enormous surprises. North America is similar to what we have seen in Eurasia, but there is one nice thing. North America, from the beginning of the cooling at about 15 million years ago and until the Pliocene, is drier than Eurasia by this ecometric. And that sets up a very interesting situation, because that means that in terms of what the future holds for Eurasian animals is already there in America. So when horses come from America to Eurasia, they find the past, and the past is always nicer than the present. So they they explode. Camels also, but camels not quite as much, for reasons that I could also speculate on, but will not. So I think, I don't know whether such broad patterns have any implications for human evolution, but I think the patterns are there and that they could be understood. Moreover, I think that we can also look in more detail at what is happening to communities made up by all these evolving lineages under various kinds of pressures and in interactions. And I select the Picarmian paleobiome. It was a kind of savanna-like biome, widespread in Eurasia in the late Miocene, after which it very abruptly disappeared. And there is the question, where did it come from and where did it go? Here you can see time slices showing 8 million years of development of this biome based on taxonomic similarity. So it's a little counterintuitive. What I want you to watch is the blue color in the middle column there. Ignore the reds and the yellows that your brain wants to focus on. And we see this biome coming in from the east, spreading increasingly westwards. About 8 million years ago, it climaxes, then it begins to fall apart again and retreat, and it's gone. Again, I will not now speculate on why this happened in any detail, but I will show you a couple of very simplistic images related to paleoclimate modeling that we did in order to understand what is happening. These are anomaly maps, so they're compared with the standard run, the control run, that is essentially the present day. And the upper one shows air pressure, and it shows this very much larger than today high pressure system sitting under this Parathetis Sea, the Black Sea, the Caspian Sea of today, were once much larger. Parathetis system, we have this. And then you equally see in the lower image westerly winds coming in from the ocean, much stronger than today, penetrating deeper into the continent. And when you look at this, you see that that is exactly what the teeth tell us. We have blue colors up where the westerlies are penetrating into this area, and we have red colors in present-day Anatolia, Turkey, where that high-pressure system is sitting. And this high-pressure system, we think, is what was driving the development of that biome, an open landscape with these large animals adapted to those kinds of resources, which are, I mean, it's a good place to be. There's plenty to eat there. It's not at all bad. And when the Pliocene comes and things become more humid and nicer again, they go, because the forests return and this is fragmented and there's no place to be for animals adapted to that. That's my guess. I've been talking about rainfall, but of course these animals are not adapting to rainfall, nor to temperature really. They are adapting to the consequences of those things, such as productivity. And it turns out that we can actually estimate something like temperature from dental ecometrics, as well as rainfall. And by plotting those against each other, we can resolve the major biomes of today's world. So I only labeled a few to give you the idea. Go from rainforest down in the left corner up to deserts and shrublands, and then tundra up in the cold there, and then all the other biomes in between. And when we take this to the past to look at paleobiomes, something very interesting is observed. Namely, that we have the Sansanian chronofauna, the middle Miocene classic assemblages of Europe, Eurasia, sitting in a part of biome space that is not really, it's sort of in between the biomes of today. And when we move up to the late Miocene, it's still rather like that. We are moving towards drier, as we would expect to be moving, but we are still, very much of it is in between, where we have no real biomes today. Now, our proxies could be wrong, or at least we could be misinterpreting what we see. I don't know. This is work just out and only a first try. But I think this is interesting. There's an interesting possibility that we talk about anomalous communities, the past being different from today. I think there are ways we can quantify that, and I think this is one possible way of doing that. So, yes, there has been a long debate of what exactly is the relationship between climate and evolution. Darwin was very much of the opinion that the organism is more important than its environment in evolution, and I think this has been, until recently, a very common view. There has also been the view represented by our friend John Alroy here, for example, that for something else, something that is nothing to do with climate, is driving evolution. And in fact, a decade ago, Alroy and others made this extremely strong statement, wondering why ever anyone could ever have thought that climate has anything to do with evolution. So I will now go into this again as briefly as I can. Just in case somebody isn't familiar with the concepts of the Red Queen and the court jester, I will remind you the idea of this royalty is that the Red Queen says you have to run as fast as you can to stay in the same place. That is, evolution is driven by competition. The court jester says, well, the world changes randomly and you just had to cope. And then there has been the idea proposed by Tony Barnowski that there is a scalar effect, the Red Queen operating at short time scales and small geographic scales, and the court jester at larger ones. And this is kind of an interminable debate still going on. I decided to look at my own work to see if I could decide how this works. And it is very obvious, actually. The Red Queen drives. No question about it. When we see animals evolving, body size increase, and we see what the context of that is when one taxon replaces another one. The larger ones are also more specialized. They have smaller geographic ranges, they have shorter durations in time. It's very good evidence that they are actually ecologically more specialized than their ancestors. It has nothing to do with the environment, they are just specializing and therefore getting larger. Pure Red Queen. Yeah, so that's just what I told you. But then I remembered something. We published something else based on the very curious observation that small mammals, large mammals, genera, they are about 5 million years in duration on average with some spread. Small mammals are different. They have the same distribution as the large, but then they have a secondary hump at about 12 million. There are some very long-lived mammals in there, and we were wondering what they were, and when we looked at what's contained there, we realized that they were all animals that show some kind of sleep or hide behavior, slow behavior. They protect themselves from the worst part of the year. That is that harshest part of environmental forcing, that they somehow escape. And when they do that, their turnover rates drop drastically. And we were not able to compare slow with non-slow. All we could do for the fossils was compare large with small. And of the small, only some of them are these slow, but of the large, hardly any. And the difference between them is that the small mammals turn over at half the rate of the large ones. So more than half, much more than half, in fact, of evolution is actually environmental forcing, climate, if you like. So the court jester drives. Case closed. So is this really a good question to ask? I mean, of course, few people have denied that both drive. And I think, no, it's not at all a good question. We can get rid of this royalty. We just need to ask, how does it work? And how can we know? And I come to my suggestion that Peter asked speakers to make. What should we do? I think we should look at traits. Traits bind everything together. They are detectors formed by evolution, optimizing them to the environment, the physical environment, the biologically mediated environment, the biological environment, and they reflect things like community composition, geographic distribution, and even, I think, coevolutionary relationships, at least potentially. They are scalable. You can apply them to individuals, populations, communities, meta-communities, and you can stack them over time, as indeed I showed you in a couple of examples, and thereby you can start to make sense of what happened. Exactly at what scales this works, I'm not sure. At the global scale, very obviously. At the regional scale, I think what I showed you about the Picermian chronofauna, I think the scale of the Black Sea is still reasonable. I think the scale of the Turkana Basin very likely is also reasonable. And this is something I would like very much to try. And that is all. Thank you very much.
Thank you, Mikhail. So we have plenty of time for questions. Anyone want to lead off? Mark? Thanks, Kat.
Thank you. I was curious, Mikhail, you didn't put generation length into the geological time, so I was really surprised that not only do small mammals turn over at the same rate and then at half the rate, but when you think about it in terms of generations, they're actually doing it at a 50th of the rate of a large mammal. I wonder, you've done a lot of work on generation time and development. I wonder how you see that factoring into what you would have expected and what you're actually seeing.
Extremely reasonable question, and I'm puzzled by this. I'm a co-author on a recent paper that argues rather the opposite, and I'm not clear in my mind why that should be. Potentially, of course, there's no question. Generation length must be one of the ultimate regulators of evolutionary rates, but I think the evidence that I've seen in what I just presented to you and in looking at such data generally is that the small mammals are generally rather slow to change, and that there is a very strong ecological control on this. So that's, yeah, I think, well, if you really need an answer, I don't think generation length actually usually is anywhere close to limiting evolutionary rates.
Paul?
So one of the puzzling aspects of evolutionary biology is actually predicting traits, not just hindcasting traits or looking at patterns of traits in the past, but predicting how traits will emerge. Do you see any hope for this? Do you see an escape valve so that there's a new way of thinking about how animals, for example, in your case, evolve?
I haven't given this very much thought, but trying to think on my feet, I think we probably should be able to do that. If it's true what I told you, that traits are the result of tuning to environmental conditions, then if we can predict the environmental conditions, we should be able to predict.
So it leads very much back to Alroy's concern, which is the selection aspects, right? So are the selections mostly neutral, or are they really to an advantage to a specific environmental condition? Are they localized, or are they more generalized? And so that brings you to Cope's rule. That's the conundrum of why do animals get larger, for example. There's no easy solution for that, as far as I can tell.
Oh, I very much agree. There's no easy solution there. But there are strong signals there. The overwhelming majority in the case I showed you for Cope's rule was that the animals were actually, I mean, it looks like we are seeing a radiation from more generalized ancestors to descendants that are more specialized and that don't even reach their peak before they branch out again. And so such patterns, I think we can infer, but it's not, no, I would never claim that it's an easy solution.
Okay, do we have some follow-on questions? All right. I mean, one of the things that I wanted to raise was just this idea that if you're, if crown height gives you such a good map globally, and that really was impressive, and particularly where the human settlement is and how that disturbs the pattern. But if if it's that powerful of a diagnostic, do you think if you are what you eat is also recorded in chemistry of the teeth? Do you think that would be an equally powerful way of mapping the ecosystems? And this sort of gets to a point that we were talking with Turi, is that there are, when you go back in the fossil record, there are some things that, for all the world, look like grazers.
Yeah.
But their isotopic composition would tell you that they're actually not.
Oh, I think there's much more to this than grazing. In fact, I think when you look at the time series of increasing crown height and opening up of environments, that has originally very little to do with grass and very much to do with open habitats, dust load on the vegetation, quality of the vegetation, the need to eat dead vegetation during part of the year, and such things that I don't think we understand extremely well, but they all work in the same direction. Grass is part of it, but it's only part of it. And in fact, it seems to me, I haven't published on this, I'm just talking about impressions now. It seems to me that committed grass eating comes in very late on all continents that I've looked at so far, except in a few groups that specialize early on
very
grass.
Very late as in late Miocene, or
later than that.
Based in the Pleistocene?
Early Pleistocene? Even as late as that, maybe in some cases. In Europe, I would say you get the first committed grazers in the latest Miocene, and then sometime later for many other areas. But this is impressions. Yeah.
So I knew this would generate a question from Naomi. So Naomi?
So just to follow up on that, obviously, so grappling with, if we had a perfect fossil record, if we had a perfect fossil record, which I know is a big if, but let's just say we did, in terms of response time, how quickly do you think that the morphology of the teeth would respond to climate change? So for instance, if Peter says he's got something at a couple 1,000-year intervals, right? If we did have the record, what would we be able to pick up in the morphology? How quickly do you think these communities would be actually responding?
Oh, almost certainly nothing. I mean, the evolutionary response, by and large, is very slow, I think. What we see is not the evolutionary response. What we see is species shifting their ranges in response to change. So the mean mean hypsodonty, for example, changes, not because animals are getting more or much less hypsodont, but because they're going away and others are moving in. Those are the fast responses we see. And then the other fast response that we have at our disposal is tooth wear, which is independent, at least almost independent of the underlying morphology. That changes with diet.
But so even if there's not an evolutionary response, there would be a distribution response that you think that we would see instantaneously in the geologic record?
In the geologic record, in most cases of the kind of resolution we have, it would look, I think, instantaneous. I would not expect there to be delays. I know that there is the idea of substantial delays out in the literature, and I think when you talk about speciation, rebuilding diversity after major perturbations, such things, there have to be delays, but in this kind of mundane, day-to-day climate change, I don't think we should expect delays.
So we have a question down here, and I believe one as well.
Okay. Mikhail, same question, phrased a little differently. Mesoware map versus hypsodonty map.
Mm-hmm.
How do they relate?
I haven't done a mesoware map, but conceptually, mesoware is diet. And as Peter well knows, about a year ago, we were in a meeting where we discussed this at great length, and it's been a bit of a mystery, and in fact, not a small worry to me, that hypsodonty and mesoware do not have a one-to-one relationship. So how can it be that hypsodonty, which is responding to tooth wear, and mesoware, that is a description of tooth wear, do not match? And we're now beginning to understand why that is, and it's to do with particle size distribution and such things. But the outcome of that is that mesoware really does pick up food. That's your grass. But hypsodonty picks up much more. It's a much broader environmental detector. It's basically openness of landscape and rainfall or humidity, effective humidity as perceived by the herbivore.
So my question. My question, I guess, relates to trying to put the observation about the rates of change, environmental change, for example, to answer maybe the first two questions. If the generational effect isn't pronounced, maybe it's because the rate of change that you're sensitive to when you look at is gradual change, for example, teeth morphology or something, versus rapid change. And if you have rapid change, like you say, it's not the slow changing of a group of species, it's more a shift from one species to another, and that may be hard to predict, which is why the prediction is difficult.
Yes.
So I guess my question is, how does the rate of change environmentally affect, for example, its effect on evolution?
Yes. Well, I think that really depends on where we are on the scale of rates. I think as long as rates are relatively slow, I mean, until you hit some kind of limit to the evolutionary response, I don't think there will be an effect. Faster change will drive faster evolution, but then you start to hit some limits where the animal is not able to change either the generation length ultimately or something before that, like constructional limits to increasing or decreasing size or whatever you have. So I'm hesitant to give a general answer to your question.
It just seems like most of the examples are relatively slow changes.
Yes, I think in this sense, at least what I was talking about could be considered slow change.
Okay. Thank you, Mikhail. Okay, our next speaker is Rowan Sage, and Rowan and I met up in Toronto. I gave a talk up there last year, and we had a little sidebar conversation after my talk, and I realized that he's someone who knows a great deal about the emergence and the importance of C4 photosynthesis and its geological history. And since much of what we're talking about, particularly for Africa, revolves around grass and the photosynthetic pathway and its isotopic signatures, it makes very good sense for us to have Rowan here with us.
Thank you, Peter. So I'm a C4 guy. My work has been the physiology, the ecology, and the evolution of C4 photosynthesis, and that's where we've been focusing. And what I'd like to do today is talk about some of the wonderment I've had about what C4 means for other animal systems, in particular the evolution of our species. And so it's going to be a bit of a mishmash of my work and stuff that's in the literature. But because C4 plants are, we think, largely arising from the declining atmospheric CO2 that's been postulated for the last 30, 35 million years, the question of whether C4 have critical implications for human evolution, did they facilitate human evolution, quickly leads to, was low CO2 a facilitator of human evolution, operating on the one hand through the evolution and success of C4 plants, and then on the other hand, as you'll hear elsewhere, through the climate system. So to get on right into it, the hypothesis, or perhaps the speculative thesis I want to address, is that features unique to C4 grass vegetation facilitated the origin of the traits that characterize the genus Homo. And you've heard a lot about the savannahization hypothesis, and what I'm proposing is it's not just savannas, but it's actually C4 savannas with traits unique to the C4 biomass that in turn facilitates the evolution of the hominids. And so I have no answer to this, but I want to propose these ideas as a hypothesis to engender discussion, feedback, criticism, and elsewhere, just in order to stimulate our thinking possibly in new ways. So the logical connection shown here, I'll just quickly go through it. Declining atmospheric CO2, there's good evidence that this has happened over the last 40 million years. In plants, it will increase the process of photorespiration. It also leads to aridification, which will slow plant growth down. Both of these processes is going to slow the growth of the C3 vegetation across the planet. But in particular, as I'll briefly discuss, this leads to the evolution of the C4 flora of the planet. And there's good evidence that this link is quite strong. You then have the spread of the C4 graminoids. Graminoids are grasses and sedges. And in time, with reduced plant performance and increasing ecological success of the C3 graminoids, you end up with mixed C4 mosaic communities, and we see these arising in the late Miocene. And then by the Pleistocene, you've had the expansion of quite widespread C4 grasslands, and based on the savannahization hypothesis, you end up with hominid traits such as bipedalism, distance running, and so on. And I'll discuss at the end of my talk how C4 grasslands in particular may have facilitated the rise of some of these hominid traits. So three themes I'll be looking at is low CO2 and the origin of C4 photosynthesis, the question of whether C4 grasses are essential for open landscapes in semi-arid to moist climates of low latitudes. There's some good ecological work that indicates they may be. And then whether the C4 grassland produces a special niche that favors or facilitates the evolution of hominids. So a little background on C4. C4 photosynthesis is a specialization of structure and physiological function to create a CO2 concentrating mechanism that overcomes the limitations associated with high rates of photorespiration, which come about in low CO2 and warm climates. If you take a C3 leaf, which represents about 250,000 species on the planet, the photosynthetic anatomy, or the photosynthetic cells, are all fairly undifferentiated. And within each cell, you have the same photosynthetic biochemistry, what we call the C3 cycle, based on the number of carbons in the first fixation product of photosynthesis. In C4 plants, you have a specialization of structure where you have the enlargement of the bundle sheath, very small in C3, into this big poofy set of cells. And in here, you have the biochemical reactions of C3 photosynthesis localized. Surrounding these cells in sort of a wreath-like manner is what we call the mesophyll. And here you have the upregulation of a C4 metabolic cycle that acts to first collect CO2 coming in from the atmosphere and then concentrate it into this internal tissue where rubisco and the biochemistry of the C3 cycle are localized. This raises the CO2 level up to very high levels so that photosynthesis can operate very efficiently. And here's the biochemical cycle. I just won't go through this. I won't go through this because it's very detailed. But the C4 cycle here, operating in the mesophyll to the bundle sheath, the key steps are carboxylation by an enzyme PEP carboxylase, which is upregulated, decarboxylation of a 4-carbon acid, hence C4, in the bundle sheath tissue, which releases CO2, raising the CO2 level up to levels about 10 times or 5 to 10 times the current atmosphere, which allows this C3 photosynthetic reaction to operate very efficiently and thus boost photosynthetic productivity quite a bit. The consequence of this for the photosynthesis is shown in this graph, where we have the photosynthesis rate, what we call net CO2 assimilation rate, as a function of atmospheric CO2. And this is for a typical C4 plant, a typical C3 plant, and at CO2 levels equivalent to the current or below, that is in the past, you see the C4 plant has a much higher photosynthesis rate than an ecologically similar C3 plant. And the ratio of C4 to C3 photosynthesis, you see, gets quite high as you go to lower and lower CO2 levels. So in the CO2 of the recent geological time, we're talking about this sort of situation where the benefit of C4 photosynthesis can raise carbon gain by 50% or more in a warm climate. So C4s, there's 7,500 C4 species, but they account for 23% of the primary productivity. 250,000 C3 species account for 75% of the terrestrial primary productivity. Much of this 23% of this terrestrial NPP due to C4s is in low latitudes, in grassland and savanna systems. The C4s are largely herbaceous. 75% are grasses or sedges. There's a few weedy dicots, and there's a handful of desert shrubs which become important in Central Asia and parts of Australia, but not in too many other areas. C4 requires warm season. It's most productive in environments in low CO2. It's not most productive, but it has a high relative productivity to C3 in low CO2. Very water and nitrogen use efficient, which affects in turn many of the plant traits that would be important for animals. And oftentimes it's pyrophytic. It does very well in a fire situation. By contrast, C3s are all plant life forms, canopy trees, small trees and shrubs, herbs, and also including grasses. All terrestrial biomes have C3 plants, low productivity in low CO2 atmospheres, less efficient in terms of water and nitrogen use in low CO2 atmospheres. If you raise the atmospheric CO2, the C3s tend to do better than the C4s because they no longer experience the limitations associated with low CO2. So if you look at a landscape like this, the grass vegetation at a low latitude, 90% of it or more is going to be C4 vegetation. The woody vegetation is going to be almost exclusively C3 vegetation unless you're in Central Asia where you get some of these shrubs. And then as this cactus here is, sometimes you have cam vegetation, which we won't focus on today. So here is the CO2 record, and so I'll first focus on CO2 and the evolution of C4. And a summary of the evidence is shown here. Here's the CO2 record coming down to about 25 million years ago. This is based on proxy evidence, and some of this proxy evidence is controversial. But this is the best evidence we have at this time, and it's also supported by various biogeochemical models. Now, with respect to the evolution of C4, we have very good phylogenetic evidence that pegs the initial evolution of C4 sort of in this period here at about 30 to 25 million years ago, at a time when the CO2 levels begin to approach current values. Isotopic evidence clearly shows the first C4 plants on the landscape at about 20 million years ago. The earliest I know of is 23 million years, and it accumulates as we come into the middle Miocene. The first macrofossils of C4s are about 12 to 14 million years old, and then by 5 to 10 million years ago, we have the big isotopic signatures for widespread C4 expansion, forming savanna-like grass tree mosaics. And then in the last 2 to 3 million years, we have widespread evidence for the expansion of open grasslands, which will be discussed by others at this meeting. So we have here, over this period of the last 30 million years, the first phase of origin, ecological expansion, and then dominance. So the first thing I want to do is just focus on the phylogenetic evidence in support of the early origins of C4. I seem to have, okay, and that's shown here. These are the dates for the estimated origin of various C4 lineages. We now know that there's about 65 to 70 independent origins of C4 photosynthesis. About 24 of these are in the grasses, 6 in the sedges, and the rest in the various eudicot groups. And taking the molecular phylogenies of these various lineages and using various assumptions associated with the divergence rates of the genes in the molecular phylogenies, we can estimate the time at which the branches appeared within the phylogeny. And with the C4 groups branching off from C3 ancestors, we can have an oldest date, an oldest estimate, and a youngest estimate. And when you do this for the 56 lineages where we have enough evidence to do a molecular clock analysis, you find that all of them are 30 to 35 million years of age or younger. And so here's our CO2 proxy estimate coming down to about current levels or near there at about 25 million years ago. And you see the C4 lineages appearing after this time. So this is strong circumstantial evidence that low CO2 was a precondition or a driver, perhaps, of C4 evolution. But perhaps the strongest evidence for the role of CO2 in the evolution of C4 comes from physiological evidence that we get from existing groups. And if you consider this and you accept there may be a role for CO2 in C4 evolution, the question becomes, how does low CO2 promote C4 evolution? And this has been the main thrust of my research. Now, to give you a little background on this, the problem C3 plants have in a low CO2 environment is instead of doing photosynthesis, here where CO2 is taken up and made into sugars using light energy in the form of ATP and NADPH, oxygen in low CO2 environments can instead be taken up by this enzyme rubisco and fixed into this molecule phosphoglycolate, which has no use to the plant and is toxic if it accumulates. Phosphoglycolate has to be scavenged back to PGA, which can be made back into RUBP to go back into photosynthesis. But in the scavenging process, previously fixed CO2 is released and can escape into the atmosphere. And this process of scavenging the carbon in phosphoglycolate requires energy, and we call it photorespiration because oxygen is taken up and CO2 is given off. In a sense, you might think of it as anti-photosynthesis, and it can be a very debilitating process for plants under the environmental conditions that promote it. This graph shows the environmental conditions that promote photorespiration. We have here photorespiration as a ratio of photosynthesis. Here we have chloroplast CO2 levels inside the leaf, and here we have temperature. And what you find is that at high CO2 levels, except at perhaps really high temperatures, photorespiration is suppressed. It's not a major biological issue, and photosynthesis in C3 plants would be fairly efficient. But if you come to low CO2 levels, and these circles show the low CO2 levels that you would have in the leaf at current atmospheric levels, the photorespiration rate becomes quite substantial by the time you rise above 25 to 30 degrees. If you go into a dry environment where the stomate's closed to save water, the photorespiration rate then goes even higher because there's less CO2 inside the leaf. And so photorespiration could be inhibiting photosynthesis above 30 degrees in low CO2 environments of the recent past by 50% or more. Okay, so on the one hand, photorespiration inhibits the performance of C3 plants, but on the other hand, it creates an enormous amount under high temperature conditions of this thing called photorespired CO2, the CO2 given off as a side product of the photorespiratory cycle. This CO2 could potentially be a resource to the plants if they can figure out a way to trap it and refix it. And it's this trapping and refixing that we now have very strong evidence for as the driver of photorespiration, of C4 evolution. And through phylogenetic analyses, we can go and look at various groups of species. So here we have the C4 evolutionary lines. Here we have this number of this is the grass evolutionary line with the red showing the C4 branches. Another major group is in the caryophyls, which includes many chenopodiaceous things. And here it's scattered, showing the distribution of C4. But we can go and study each of these groups and study the C3 ancestors, the C4 progeny, and importantly, groups of species that are intermediate between C3 and C4. And using the phylogenetic analysis, we've been able to identify quite a number. And as shown in this one group, Flavaria, which has been heavily studied, we have ancestral C3 species shown here, derived C4 species shown on the branches in red, and then in the blue and the yellow we have C3, C4 intermediate species, which we've been able to study and put them in a phylogenetic context and look at the progression of C4 evolution. And so here we have a C3 ancestor in Flavaria, that's right down here. Here's a C3, C4 intermediate species, and compared to the C3 ancestor, you begin to see the appearance of Cran's anatomy. You see enzyme shifts shown by this blue stain in an immunolocalization study from the mesophyll into the bundle sheath. And then we can study the progression as we go up to this species shown here, where we have the full C4 anatomy with that full wreath-like Cran's anatomy. And the phylogenetic analysis has allowed us to identify at least 10 lineages where we have C3, C4 intermediates to look at, so we can do a comparative analysis and ask questions of, do all C4 evolutionary forms arise through the same sequence of steps, or are there different pathways to C4 evolution? From the biochemical, physiological, anatomical studies, we've come up with a model of C4 evolution, and for all the lineages, it's pretty consistent, shown here in this pyramid diagram. And I won't go through each of the steps, but I'll highlight some of the general ones shown here. The first phase of C4 evolution is rearrangement of the leaf's anatomy, initially to save water, which then creates a Cran's-like condition called proto-Cran's anatomy. This sets it up so that you can have an adaptation where a key photorespiratory enzyme is relocalized into the bundle sheath, giving us the ability to concentrate CO2 and make photosynthesis more efficient by recapturing photorespired CO2. And in the process, you get full Cran's anatomy during this phase. And then the final phase that we see is once you have the structure in place, you get the layering in of the C4 biochemical cycle. The key take-home message here is that only under conditions of high photorespiration do you get the sequence of steps leading to the production of this photorespiratory CO2 pump. High photorespiration pretty much requires low CO2, and it's facilitated by aridity and high temperature conditions. The photorespiratory CO2 pump, just to quickly show you how that works, this is the photorespiratory cycle in a C3 plant. It all operates within a single cell. Phosphoglycolate is converted to glycine, which goes into the mitochondria, where an enzyme glycine decarboxylate it, releases the CO2, produces serine, which is made back to PGA. The CO2 then escapes the leaf, causing the reduction in photosynthetic efficiency. Now, the ancestors of the C4 plants, and this is shown in those 10 lineages, what they do is they concentrate the mitochondria with glycine decarboxylase into the bundle sheath. They enhance chloroplast numbers in the bundle sheath, and they eliminate glycine decarboxylase in the mesophyll. This forces all the glycine into the bundle sheath, which is an internal layer of cells. And then when you have the photorespiratory CO2 released by glycine decarboxylase, that is trapped within the bundle sheath, and all the rubisco and the chloroplast within the bundle sheath can refix this with high efficiency. And it's this process, photorespiratory CO2 concentration, that we consider is the bridge to C4 photosynthesis. And again, it only happens in an environment where you have high photorespiration. Now, changing directions. So we evolved C4 photosynthesis in response to conditions leading to high photorespiration. The Earth has become depleted in its atmospheric CO2 in the last 30 million years. What is the consequence of low CO2 for the spread of C4-dominated systems? And in particular, are C4 grasses required for open landscapes in semi-arid to moist climates? So C4 photosynthesis, as I mentioned, is more productive in low CO2. It's more water and nitrogen use efficient. If you have a disturbance, a fire, or a massive animal herbivore event, the C4 plants, the C4 grasses, can recover quickly from this. C4 grasses are also very strong competitors against C3 grasses, C3 vegetation in low CO2. C3 grasses, by contrast, don't have any special photosynthetic advantage over C3 woody species. But the C4s do. And this is a graph that Jim Erlinger and Terry Serling and so on produced a few years ago, showing the relationship based on physiological properties, where with the CO2 level going up, you get more C3 vegetation favored. If the temperature goes up, you require more CO2 in order to cross over to having C3 vegetation favored. Now, in a warmer environment here, one of the interesting things we've had in the last 150 years is CO2 levels have gone up from about 280 to now close to 400 ppm. And so for many parts of the Earth, corresponding to areas of South Africa, the southwestern US, Australia, we've had the CO2 level shift from C4 favored to C3 favored. So the natural experiment is already undergoing, and we can look to see whether we're getting a shift from the C4 dominated to a C3 dominated system. And I'll show you the results of some of that at the end of this section. But just to begin with, controlled experiments of woody C3 things from the African savannas find that as you raise the CO2 level, particularly as you go above 280 ppm, you get a big boost in the growth rate and the biomass produced by things such as Acacia carew and Terminalia seracea. In particular, the resprout rate following a simulated fire takes off as the CO2 level goes. So CO2 is now recognized as a strong fertilizer of C3 plants, especially woody C3 plants, following a disturbance event. And Bill Bond, who's responsible for a bit of this work, has done a nice experiment where they've looked at root growth under different CO2 levels. So here's 180 ppm, the CO2 of the Pleistocene. You see the roots do very poorly in Acacia carew. At higher CO2 levels, you get a very big, dense root mass. And in a semi-arid environment, this is critical because it allows this woody thing to establish, to get an abundant source of water, to get below the C4 grass competition, and thus to be able to establish. For the roots to get to below the C4 grass competition. An interesting experiment done some years ago by Hiram Johnson's group in Texas, they took Texas savanna vegetation, grew it at 200 or 270 ppm CO2, and they looked at the C3 to C4 performance ratio, or C4 to C3 performance ratio. At 200 ppm, the C4 biomass, a mixture of species, produced 15 times as much dry matter as the C3 biomass did. At 270 ppm, you see that difference has dropped to 3 ppm, and at current CO2 levels, more recent experiments show that they're about equally mixed. So here we have then CO2 levels naturally increasing from 305 ppm in 1925 to 390 ppm today. And over much of the Earth, as shown in this picture from South Africa, you've had an enhanced woodiness. So this grassland system has largely filled in recent decades. And you see the same thing happening in Texas, in much of Africa, much of Australia, and so on. And so we can have then ask the question, if CO2 levels hadn't declined to low levels, would we have been able to evolve or have a system like this, which we know existed in the last century at low CO2 levels, such as in West Texas? Would this system instead have been something like this? And would hominins have evolved in a system like this, a dense brushland, as opposed to a much more open C4-dominated grassland? And that's, I think, an important question for us to discuss. Finally, what does C4 vegetation mean? So you have C4 grasslands expanding across the Earth. What kind of nutrition does this present for evolving hominids? C3 plant resources are quite diverse. You have fruits, you have nuts, you have berries, you have tubers, grains, bark, leaves. And so you have a diverse food set, but as we know from today, these can be relatively sparse on a C4-dominated system where 90% of the NPP is C4 grasses. In a C4 system, however, the amount of food presented is fairly limited. Fibrous leaves, fibrous roots, small seeded grains, which are easily shattered, small, often very fibrous rhizoids. So if hominids are moving out into the open savanna, what are they going to be eating? The foods of a C4 are relatively low quality, but, however, they're very abundant in these relatively high productivity grasslands. And even today, the consequence of the C3 versus C4 difference is seen in our diets. So right now, there are hundreds of domesticated species that make up our diets. For example, all the fruits, all the nuts, all the tubers, all the squashes, all the legumes, all the root vegetables, nearly all the oil crops are all C3. The C4 things that we've bothered to domesticate is relatively sparse. A couple dozen grain species, most of which are not very popular. Maize is the huge exception as the one that's widely used. But the others are millets, phonios, sorghum grain, and these are all annual plants. None of the perennials produce grain that we use. Sugarcane has been domesticated for sugar. The vegetables, the leaf-eating things, there's only just a small handful of things we directly consume, and none of these are grasses. They're amaranth and Portulaca oleracea. So the C4s don't provide humanity with a lot of direct food material for us to consume. The situation with maize is fairly specialized because for some reason that's produced a very large grain. Why so few C4 foods? Most of the C4s are graminoids. They tend to be small-seeded, and there's physiological reasons associated with this. The seeds are easily shattered. They tend to produce relatively few, often small storage organs, often very woody storage organs, not like big sweet potato tubers. The leaves are often very fibrous, and importantly, the leaves are low in nitrogen. The C4 system has a nitrogen use efficiency that's 2 to 3 times that of a C3 system. What this means is the C4 plant produce a lot less nitrogen in the leaves because they don't need to use nitrogen so much to meet their carbon needs. This leads to low leaf protein levels. And then to top it off, much of the best protein is housed in the bundle sheath, which is a fairly fibrous tissue sheath that is difficult for us to digest. And so you have low nitrogen, and much of it is inaccessible to humans. And so this then leads to a condition where much of the C4 biomass is of low quality, if at all digestible. Now, certain animals, such as the bovine ruminant, are well specialized to eat C4 biomass. They have the very large forestomach, where you get a lot of bacterial action breaking down this material. And you have the rumination, where you regurgitate and remasticate this very tough material to break the stuff out of the bundle sheath. And then when you look at other C4 herbivores, such as horses, you see a very large, well-developed hindgut where you can have extensive bacterial action and extensive time to work through and digest the C4 biomass. By contrast, in chimps and humans, and presumably our common ancestor, we have a relatively simple digestive tract that's not going to be able to handle high-fiber C4 grasses. Young, new shoots of C4 grasses, perhaps, but to address the situation of what humans are going to be eating on the C4 grasslands that may have opened up 2 to 3 million years ago, we have to address the issue of what is the nutritional quality presented by the C4 vegetation. And so the C4 niche, the C4 grassland niche that humans may have spread into, is probably not going to be heavily found in the biomass of the plants, the C4 vegetation. But because these are so productive, those grazing species that are specialized to digest this, the zebras, the wildebeest, the others, well, they're a very valuable resource that humans can exploit out on these landscapes. So when we talk about opening up the landscapes, you may be talking about an excellent niche if you can be a hunter or a scavenger. Not so much if you're an herbivore for a small animal like the hominids. And so the possibility then is that human evolution was driven by our ancestors going out and taking down the secondary producers who could digest the C4 vegetation. And of course, if they're going out onto these big open landscapes, they have to deal with these kind of guys who would easily take down a relatively small, slow-footed animal like an early hominid. And so a modification then on the savanna hypothesis is that hominid exploitation of these open C4 niches could have been hominid exploitation of the C4 herbivore resource. And then you can ask, what traits would this require? Well, for example, upright stature with free hands, the bipedalism aspect, endurance running, coordinated hunting and scavenging in groups. For defense against the felids, you have to have coordinated defense, probably some kind of weapon activity, whether it's thrown rocks or sticks. And then also extensive communication skills and enlarged brains. And so this is something where I think, as we discuss ideas like the savannahization hypothesis, we have to discuss the unique features of the C4 biomass. Okay, so I'll stop there. And here's a few research questions we might talk about. What is the composition of edible plant species on C4 grasslands where hominins evolved, that is, edible from a human standpoint? What is the extent that could hominids have directly exploited this C4 biomass? Could they go out and eat these young grasses or something? And is a C4 isotopic signature we see in hominids a sign of secondary consumption, that is, scavenging or hunting? So that's where I'll stop. Thank you.
Okay, we have a few minutes for questions. Kate.
Hi, Rowan. Good to see you again. Kate Freeman from Penn State. I'm curious about two elements of the CO2 link to the expansion of the onset of C4 emergence, and of course you didn't quite address the expansion later. And I'm curious if you could speculate.
I figured you guys have better data on that than me.
On that, and in particular, I'm curious about the link to aridity and fire prevalence. And I'm wondering if you could speculate on those.
On the expansion?
Yes.
Okay, well, the fire link is the idea that woody vegetation often is very persistent. If it can establish, it can hang out and take advantage of whatever good opportunities there are and gradually work its way above the grass canopy. Fire would knock that woody vegetation back. For many of the savanna species, then they can resprout and come back. But after repeated fires, that gets more and more difficult as the mortality rate increases. For woodland species, which tend to be less fire adaptive, the fires are lethal. So a key issue is when you have higher CO2 levels, these woodland species can establish faster and get their roots down and get their canopies up above the fire kill zone. And once they can get up above the fire kill zone, then they can shade out the C4 grasses, which are very high light dependent, and gradually then capture the site. And so the idea is that low CO2, the recovery rate from a fire or the establishment rate of a C3 plant, woody plant, is so slow that they're most likely going to be killed by a typical fire return interval. Plus, when it's low CO2, the C4s can just directly suppress a certain fraction of this vegetation through direct competition for water and nutrients. So in a severe drought year, you're going to have a certain mortality of the young woody plants. But once they get their roots down below the woody vegetation, the mortality rate from competition for water during a drought year goes way down.
Is this what happened in Australia 45,000 years ago, when McGee and Miller have shown that isotopic evidence that there's a big shift? I can't remember which way.
I think it's a shift towards a C4 system.
Yeah.
45,000 years ago, you get a big...
when the man came in, so there...
Yeah, you get that big charcoal signature, so it looks like people have thought about the fire signature going way up. The problem is I think the evidence is for human introduction into Australia about 70,000 years ago, and that 45,000 year signature in the charcoal record is basically the end of the carbon-14 dating window. So it's possible that that 45,000 date could be variable, but the thinking is that's a human signature driving up the rate of burning.
Interesting. Maureen? Oh, Paul?
Okay. So Rowan, the isotopic signature of C4 plants, if I understand it correctly, going way back to Cooper and Lane and so on, is based on the fact that the primary carboxylase is fixed by carbonate rather than CO2. Is that true?
You mean the reason the C13 ratios shift?
I mean, that's what a lot of the geochemists use as basis for the diet, for example.
I've heard that, and I have to scratch my head. So the initial fixation by PEPCase is not a strong discriminator like the initial fixation is by Rubisco.
Right.
So, but you still have that Rubisco discrimination in the bundle sheath. So the delivery into the bundle sheath is has a very low isotopic signature. Then you get the Rubisco fixation, which has a strong isotopic signature. But in a C4 plant, the CO2 that gets into the bundle sheath has difficulty getting out, either because it's immediately fixed and is trapped there by the diffusion resistance, or it's captured on its way out and sent back in. So the C4 system tends to be a closed system.
Okay.
And that then is the isotopic signature of the PEPCase in dominates because everything that goes into the bundle sheath gets fixed except for what leaks out.
So let's follow the logic.
Yeah.
If we go up to 1,500 ppm CO2,
right,
the Rubisco would then not have as strong isotopic fractionation? So a C3 plant would look like a C4 plant?
No.
Isotopically?
You'll still have the same isotopic difference between them.
Why?
It might shift slightly. because you still have that initial fixation step of pepsiase. The bundle sheath is really sealed, both from inward or outward diffusion. There's some leakage, but the main thing is that pepsiase drives what gets into the bundle sheath.
So it's only because the closed system, you think?
The closed system is the predominant thing, yes.
You could say it's a competition between two rate limiting steps. And one is the uptake of CO2. The fractionation is small, and the viscofractionation is large. So it's sort of a competition between the two. And in a C4 plant, you pretty much the rate limiting step becomes the uptake through the membranes, or through the stomata, whatever they have, and that gives the small fractionation. Then it's almost entirely used when it gets to the Rubisco, so there's no more fractionation because you use it all up.
Yeah, that's pretty much it. I wouldn't use the word competition because these two things are in series. If they were in parallel, I would think they were in competition. So because they're in series, you get the one feeding the other, and then because it's trapped in there, you get almost complete fixation.
Your photographs of the 20th century changes in Texas and South Africa were really dramatic. And how do they know that there's no human agricultural or domestic fuel use effects on that?
There is obviously a fair amount of human effects on that. But you can go to systems, and I think I got a picture here, if I didn't take it out, where you can get away from that. So here's one for the Sonoran Desert. This isn't a C3, C4 system, but a CAM C3 system. This is in the Sonoran Desert National Park. From CO2 levels, 310 in 1935 to today, and you see the same phenomenon here of increasing woodization. You go to island systems where you haven't had a lot of human, grazing's the big one that's selected against the grasses, and you've had the same thing, woodization. They've tried to do experiments where they recreate the savannas, take out the grazing, manage the fire, and so on. That would obviously be modulators of this woody versus grass. And they often find that today you get easy establishment of things like mesquite and so on in these recreated grasslands. So it's really hard to maintain them without extensive active management. And so it's not proven that this CO2 is the driver, but there's increasing evidence and an increasing sense that while CO2 wasn't necessarily responsible initially for loss of C4 grasslands, grazing, for example, or fire suppression would be, today it's become a strong driver because we've had this fairly substantial CO2 rise in the last 100 years. It's about 25% of the CO2 rise, but that 25% in the last 100 years is the equivalent of doubling in the next 100 years because we're down at the lower end of the enzyme substrate curve.
One more question, please.
Following up on Paul's question about the closure and the leakiness of the system, I'm curious, and I was really intrigued by the intermediate forms that you have been studying. How does the Krantz anatomy influence heat dissipation by these from the photosynthetic activity? And does that heat dissipation influence leakiness? In other words, does PCO2, is there a linkage between heat dissipation, water loss, and CO2 that might also influence leakiness?
Not too much. No, not much evidence for that. The heat dissipation is largely through evapotranspiration or convective heat exchange. And the bundle sheath doesn't influence that very much. What influences that is as you go from the intermediates to the C4s, you get the establishment of the C4-like stomatal conductance system. We don't know what the mechanism is for that. But that's where you go from a system where with a C3-like transpiration rate to a C4-like transpiration rate, and then you get a bump up in leaf temperature just because of reduced evapotranspiration. And so does leaf temperature affect leakiness? I haven't seen any data on that at this. Yes, it does, actually. When you go to low temperatures, leakiness goes up because the C3 cycle slows faster than the C4 cycle, so your concentration in the bundle sheath goes up and you get more leakiness out. At higher temperature, at least up to the thermal optimum, that's where you have your minimum degree of leakiness.
Okay, so thank you very much, Rowan.
Thank you.